The overall goal of this multimodal micro-characterization procedure is to provide a method for the location-dependent correlation of the data acquired by X-ray computed tomography, light microscopy, and scanning electron microscopy.
This method can help answer key questions in the micro-characterization field, such as failure analysis or reverse engineering of microelectronic devices.
The main advantage of this technique is that optical properties of a sample may be linked to the microstructure and even to submicrometer structural details.
The implications of this technique extends toward prevention of device failure, because optical defects or inhomogeneities can definitively and traceably be linked to structural or electrical defects of the devices.
So, this method can provide insight into microelectronic devices. It can also be applied to other structural analysis, such as characterization of composite materials.
Begin this procedure with sample preparation and CT measurement setup as described in the text protocol. Select the region of interest, or ROI, by identifying the area not obscured by the measured object during one full rotation. In the measurement window with the live image, press and hold the left mouse button and draw a red framed window. Right-click on this window's frame to open a context menu. Then, select Set as Observation Window. The frame's color will change to yellow, and the observation window